Good morning, friends. Today we are going to see Ti-plasmid of Agrobacterium tumifaciens. First, I will introduce you what is Ti-plasmid. Agrobacterium tumifaciens is an aerobic gram-negative soil bacterium discovered by Smith and Townsend in 1907 and is popularly called as natural genetic engineer. In nature, Agrobacterium tumifaciens has the ability to infect plants, mainly dicotyledons, and causes a disease known as crown gall. Agrobacterium tumifaciens possess a plasmid known as tumor-inducing plasmid of about 200 kb in size. Crown gall formation is the direct result of transfer of portion of Ti-plasmid into the plants. The general features of Ti-plasmid are as follows. It consists of tDNA, VR region, ORI sites and TRA sites. Ti-plasmid also contains genes for the synthesis and catabolism of a special type nitrogenous compounds known as opines, auxins, and cytokinins. The VI region regulates the transfer of tDNA into plant cells and lies outside the tDNA. VR genes of Ti-plasmid are required for mobilization of tDNA from the bacterium to plant cell. Once successfully transferred, the tDNA forms a part of plant genome. tDNA region contains oncogenes and opine synthesis genes and is transferred into the host plant genome. The transferred DNA is approximately 23 kb in length and is transferred into the plant genome in tumor cells. Opine catabolism regions produce enzymes necessary for utilization of opine as their carbon and nitrogen sources by Agrobacterium. The left and right border of tDNA exhibit 24 base pair unique sequence of direct repeat and are invariably transferred to plant cells. ORI regions are responsible for the origin of replication, that is, for the propagation of Agrobacterium. The TRA part is helpful in conjugative transfer of plasmid. E. coli is widely used in recombinant experiments, but Ti-plasmid cannot replicate in E. coli. So, considering these limitations, Ti-plasmid based vectors have been constructed by using different strategies. Let's see the physical map of Ti-plasmid. Now this is Ti-plasmid having ORI gene in replicator region, VR genes in virulence region, which lies outside this tDNA region. These VR genes are responsible for mobilization of tDNA of Agrobacterium to plant cells. The region from left border to right border is called as tDNA region, which consists of auxin, cytokinin and opine region. The left and right borders are of 24 base pair repeats. Vectors based on Ti-plasmids have been designed with the following properties. They do not cause the disease since they are disarmed by removing oncogenes from the tDNA causing the disease. Having the right border sequence which is required for the integration of tDNA into plant cell DNA. They carry multiple cloning sites for insertion of foreign gene intended to be transferred. An origin of DNA replication that allow the plasmid to multiply. They carry selectable marker genes, among which the most commonly used marker gene is the gene encoding neomycin phosphotransferase, imparting resistance to kanamycin, so that the transformed cells will grow in a medium supplemented with kanamycin and carbonicillin, and transformed cells will be appropriately selected. At incubation period, Agrobacterium tumifaciens infect the leaf disc through wound done during disc preparation. These discs are transferred onto the fresh shoot induction agar medium containing suitable concentration of kanamycin and carbonicillin. In this medium, only transformed plant cells regenerate to shoots because kanamycin present in the medium will not allow normal plants to grow. The antibiotic carbonicillin inhibits the growth of Agrobacterium tumifaciens. Then the regenerated shoots are subcultured onto fresh medium for proper growth and roots are induced. These recombinant or genetically modified plantlets are finally acclimatized to soil condition. Significance of Ti-plasmid. Ti-plasmid is the cloning vector used for many dicotyledonous plants and has a broad host range. When a desired gene is linked in the modified Ti-plasmid, the desired gene can be transferred to the plant genome and is stably transmitted through plant cell division. Practical applications. By using recombinant DNA technology, it is possible to produce plants which are resistant to herbicides, salts and diseases, etc. By using recombinant DNA technology, it is possible to obtain the plants with generally insecticidal activity. This reduces the application of chemical pesticides to the crop plants, and this method acts as eco-friendly approach. Hope you understood the topic very well. Thank you.